So it probably has been around forever. It's just wasn't identified as a specific disease. Some people get the heart condition, which is it's called tetralogy Fallot. It's basically a heart birth defect. If you ever heard of Jimmy Kimmel's son, has that birth defect, and so they also have liver defects. And if they also have liver defects and a variety of other defects, then it's likely that they have Alagille syndrome. And in the past, it was just identified based on the cluster of pathologies. But ultimately, with the the human genome sequencing efforts from, I guess, more than a decade ago, but yeah, well, actually a couple of decades ago. And so it's actually really interesting because Francis Collins, who is head of NIH, was actually the person who helped to clone this disease. So they identified the mutation, and that was done, I think, in '97. And so it turned out to be a mutation in the gene called JAG. Right? JAG is interesting because it's basically a receptor from one cell, and it binds to the notch. No, it's a ligand from one cell. It binds to the notch receptor on a neighboring cell and allows the cells to communicate, basically. And this pathway is just found in every animal. It's actually discovered in fruit flies, which I've worked with fruit flies before. But back to the point, so it was identified a long time ago. And back in the days of the whole promise of, if we sequence the whole genome, we'll get to find answers for diseases real quickly. The answers turned out to be mostly what causes the disease, but how to cure it has been a much more difficult road. And we're hoping now our lab is bridging one of these cures. So basically, if you're born with this mutation, it's usually you have two copies of a gene. You only need one mutation to get this disease, and even if you get this mutation, you're not guaranteed to get this disease either. And so the other interesting thing about this disease is sometimes it happens spontaneously, meaning your parents don't have to have it, and you can just spontaneously have it because of a mutation that your parents don't have, right? And so and that happens in most cases in Alagille syndrome. And what that means is you can't really genetically counsel your way out of this. You can't just say, well, if the parents have this mutation, then they just won't have a baby. It's happening without the parents having the mutation. So this disease is probably not going to go away. And this is why I said this probably existed a long time ago. And so now they're just starting to identify, as many other genetic diseases are being identified now. Although it's considered a rare disease, it's not that rare. So it's about 1 in 40,000 births. And if you imagine millions of births around the world, so each year, about 3,000 to 4,000 people are born with this. And depending on how severe the individual patients are, the effect on their life is variable. But on average, you do have this liver problem, and if you don't get a transplant, about 50% survival rate at the age of 20. So it's a really big problem. Okay, and on the research side of things, really what we know are the mutations, and what that gene is important for. Actually, we published in our lab a paper a few years ago in Nature Communications to show that without this gene, those duct cells are lost in model animals, and that's also seen in people with this mutation. But the important thing is we discovered where the source of that gene is signaling from. So the mutations in the JAG, as I mentioned, it codes for basically a signaling ligand that then signals to a neighboring cell, right? And without that signal, that neighboring cell isn't instructed to become a duct cell in the liver. and that's why there's less of it. And so a lot of people study, for example, liver regeneration, but only a handful of lab actually studies liver regeneration due to, actually less than a handful of lab study liver regeneration due to jagged loss of function, right? So if you remove jagged and you have less ducts, if they regenerate, how do they regenerate? Really, there's only a couple of other labs that study this. And so that's why we're focused on that. Other people are studying this pathway in other parts of the body. Not all of them are focused on specifically the jagged mutation. They may be studying other parts of the pathway. But all this different studies kind of come together and help us really see a bigger picture. But in our lab, what we're trying to do is really focus in on the gene that's affected. And then we've created a model where we remove that gene and then we let it come back. And the surprising thing is that when we let it come back, it regenerates, right? And so just imagine in these study animals that we study, they have absolutely no duct cells in the liver. But if we let the gene just function again normally, we were really surprised to find that duct cells start to regenerate. And then our next step was, where do these cells come from? Because other people who've studied regeneration using other genetic models, they're saying that while these other cells in the liver kind of change their identity, they transdifferentiate and they become duct cells. And that's what we were looking for probably well over a year, and we could not find that. But keep in mind, our mutation is very different from their mutational model. We actually got rid of the gene jagged itself. And so that's where kind of the hard part came in, is it didn't come from that area. It came from that cell type. They're called hepatocytes. They're one of the two cell types in the liver, okay? The duct cells and the hepatocytes, right? And so people are thinking they would change and become duct cells, and we didn't see that. And so we kept looking and looking. Eventually what we found were this new source of stem cells that are coming from basically where the liver attaches to the gut, right? So normally the liver produces all this waste, it goes through the duct and it goes through this other duct called the extrahepatic duct, which connects the liver to the rest of your gut. We found that there are progenitors there that sends out, that keep stem cells and send regenerative cells into the liver. And that paper right now is under review. So we basically reported a new source for regenerative stem cells for the liver. But that is just for our understanding. To me, the important thing for people who care about dealing with Alagille syndrome is that there is a source of regenerative cell, and that these cells can regenerate if you let that affected pathway come back. So then the question is, how can we help this pathway come back in patients, right? And I guess the most obvious thing is gene therapy, right? Can you fix the gene? But that's still not an easy thing to do. Even with CRISPR, there's going to be a lot of work. So another approach is, can we find a drug that will increase this pathway? And so for the last couple of years, we actually have identified a drug that can do this. And so when we found this drug that can increase this pathway, and we've tested it in mammalian cells. Our lab uses the zebrafish model, and it allows us to quickly answer all these questions, do some complex genetics to figure out where those stem cells came from, and actually even to create a model to find that this loss of this gene and loss leading to loss of these duct cells are reversible. But the drug that we're working on, we wanted to make sure it works in humans. So we've tested them on mammalian cells, like mouse cells, mouse liver cells. It can increase this pathway, increase those cells, get those cells, liver cells to express, to become duct-like, right? To turn on these duct genes that normally that Notch pathway can turn on. So that drug can increase Notch, increase these cells' ability to become more like ducks. And so we've tested them in mouse liver cells. We found out where the target is, what it binds to, and we think it binds. We have actually pretty good evidence that it binds to the Notch receptor itself, and then can turn on genes that are important for ducks, right? And so we've even taken Alagille patient cells. So we tried 3 different patient cells now and tested the drug, and it looks like it can activate this pathway. And more recently, we went back into the fish, and we took our mutant model of jagged, where these ducts don't form very well, and we found that the drug actually can get them to regenerate better. And of course, we're interested in how that works. We figured out what gene that drug is activating to make more duct cells, and so everything looks really promising right now. Our next thing is to test this in jagged mutant models of Alagille syndrome in mice, and then from there, we're hoping to be able to then head towards clinical trials. So what's great about this drug is that it actually targets the pathway itself. So even though we're working on the liver, what I'm trying to say is that this drug hits at this genetic disease in a fundamental way. We're actually increasing the pathway that is lost. So that jagged mutation binds to the Notch receptor, activates that pathway. What we're able to do is find a drug that binds to the Notch receptor also and activate that pathway. So what I'm trying to say is that although we're gearing and testing this potential new drug for resolving the liver problems, which we're showing is reversible if you let this pathway come back, would it be useful for other pathologies in Alagille syndrome? Will it help decrease the risk of bleeding, for example? Is this something that we're really interested in testing next? And so yeah, we're very thrilled about this drug. And it's actually kind of interesting because when we first started testing it, we actually didn't expect it to work. And initially we didn't think it worked, but we did some more experiments and we realized that it worked. And so the interesting thing is the postdoc in my lab who's working on that drug, he actually was working on that other project I was telling you about that he was so that's so interesting. As soon as he realized the drug worked, he basically dropped everything and has devoted his last 2 to 3 years working on characterizing this drug. And then that's Jonathan, and then another postdoc in my lab who's also very productive and has made many discoveries. His name is Jerry Zhao, and the other postdoc is Jonathan Matalonga. And so Jerry has been the person who's done the work on the regeneration of the liver. So he came into his lab. We told him these things can regenerate. We think we might know where it came from. He actually did all the genetic work to figure out where the source of the stem cells are, this new source, and he more recently tested the drug in the animal models of duct loss, the jagged mutant models, and found that it actually works there also. Normally anything like this can go wrong anywhere, right? But of course you got to get into clinical trials, but before you can even get into clinical trials, you have to demonstrate that it works, it's not toxic in animal models and so on. So that is an extended process that may take more than a year, maybe years to do, to just even get into clinical trials. So we're trying to move towards toxicity studies right now. So far we have injected it into mice. We haven't seen any major problems yet. One of the concerns that a lot of people bring up is, well, this pathway is so fundamental. Do you really want to increase this pathway? You're just gonna muck up the rest of the other tissues in the body. And that's where I think we have to take into account two factors. One is that you keep in mind that not everybody with this mutation has the pathology. And even if they do, it can fluctuate in life. And that actually tells us even an additional thing. The fact that it can fluctuate in life, that means things can get better. But the fact that it fluctuates also tells us that the level of jagged function, because you still have one copy left, that is probably on the threshold of what is enough and what's not enough. And the goal is to really kind of nudge it over to a level of jagged notch signaling that is sufficient, so that you're more healthy. And so the goal is not to put a ton of this compound, and that's kind of counter to a lot of other drugs that we use or we think we need. In that we sometimes, like with cancer drugs, we want to hit certain things really hard to kill those cells. Here we just want to nudge things a little bit, which is a completely different concept. We don't want a drug that is too strong. We want something that's just weak enough to just nudge it. Although we think this drug is strong, we can. The way we envision it is that the dosage would be very low, and just to nudge things over a little bit. You won't need a lot of it. The other is that this drug, because it targets the receptor, not every cell has the receptor. So that means you're not just going to turn this pathway on in every cell, all right? And the other thing is, it's not permanent either. You can remove it and it goes back down. And so we envision not using this in a persistent way and not at very high level. So we don't think it will actually be too harmful for the body. And actually, when we first tried using it, we dosed a ton of it on developing zebrafish embryos, and they did really well. Surprisingly, I thought a drug that increases this pathway would just mess everything up. But in studying this, we were learning a lot of things. For one thing, I mentioned earlier, this drug works on the receptor, and that receptor is not on every cell, so you're not going to muck up every cell, right? The other thing is that cells are actually very resilient. If you do something to perturb them, they will try to shift back to a normal state. And so when we've seen this, even in cells in culture, we can add the compound, it'll increase this pathway, but just several hours later, it'll just be kind of dulled down, so it doesn't increase too much. And so for those reasons and other reasons, we think the drug, so we're convinced that it does uprate the pathway. The question, is it going to do too much? Is it doing it, it's going to overdo it in cells? And we have reasons to believe that that's not going to happen. And even if it does, we can always reduce doses and so on. But as I mentioned, it is a fundamental pathway. We are aiming this notch agonist, which increases notch pathway, for this for Alagille syndrome specifically. But we think it's going to have many other applications. But we're focusing first on Alagille syndrome, and once that gets going, then we're going to try to test it for other applications, for other diseases, for biotechnology. There's no other. So I forgot to mention, there's no other compounds out there that can increase notch signaling. Nothing near this level of increasing it, and so it's a first in class. So I think there's going to be many more applications beyond Alagille syndrome. But I think Alagille syndrome is one of the most pressing problems right now. That's why we want to approach it first. It's actually an interesting story. It was a Friday night experiment. We got the compound. We put it in zebrafish embryos at the beginning. We didn't see anything, but we weren't looking carefully enough. We thought, well, if this compound works, our fish fluorescent model would just glow like crazy and we should be able to see it. And it's not like that, and actually it's good that it's not like that. So we didn't think it worked, and so I asked Jonathan in my lab, said, could you throw it on some of these human liver cells, since you're working on them for other things? He did it. It worked. He texted me and he was like surprised and like, oh, that's interesting. And then he tested it again early next week. It worked, and then it worked again. And I think that was when it dawned on us that we got something, and that's when he kind of started to drop his old stuff. which is still, I mean, we're working on. Of course, I have other people in the lab working on it, but yeah. So it's just what do you call it? Really unexpected to kind of bump into a compound like this. We did not think it will work. It's working out better than we thought. And so that's kind of the history of how we started to work on the compound. I think a lot of people just ignored it because for one reason or another, it's a lot of times it's this feeling like, oh, that's too good to be true, and we thought that was the case also. But we our lab, we tend to give things a chance. We test things. If it doesn't take too much to test it, we'll just test it. I mean, it's similar to our other work where we're reprogramming cells. People thought it was impossible. We had a model that allowed us to just do it without a lot of investment. So we started skeptical to begin with. But I think one of the things that you have to be careful with in science and in life in general is not letting your skepticism be an answer. Your skepticism is just to help you be careful. But if it prevents you from trying new things, especially if you have reasons to try it, then it's worth it. I think too many times we think things are too good to be true, or we assume based on our education that certain things are impossible. If you think about science that way, right, then we only would be working on things that we expect to work. And what fun would that be, right? I mean, where is the major discoveries? Because if you find what you expect, it's expected already. How amazing would that be? So I think really it's, even though you may have some skepticism in certain things not working, you may have other reasons why it might still work, or ways to get around why it might not work, and then that's where the great discoveries are. I think it's just really knowing, really challenging your assumptions, right? And because to me it's like, if you do an experiment that you assume will work and it didn't work, to me that's the beginning of something amazing, right? Because it's not working because your concept of that, assuming you did the experiment correct, of course, that means your assumption of how that works is not true. And then that means if you figure out why it didn't work and why you had a different result, then you're actually breaking an assumption, breaking a concept. And I think that's really important in science. I think too many times people are asked to write grants about things that should work a certain way, and everybody thinks, oh, you should do this experiment because according to this, this will work. Of course, everybody thought of that already, and people are doing it. How novel will that be if you're doing things that are kind of expected? But the problem is, if you're doing something that's not expected, it's easy to criticize. It's easy for people to say, oh, that's not going to work because we know this and this and this. But that may work because we didn't know this and this. And that's really where the good science, I think, the cool new science comes. You still can get great science from other ways, but I think if you really want to find something new, you gotta challenge these assumptions, these skeptics. Okay, to study Alagille syndrome, actually, I can take you back to my robotics day in high school. So actually, I was talking to my son about this the other day. I told him, so when they first proposed the challenge, the rover challenge, the first thing they tell you is, okay, what the challenge is, which is, for the first year I was doing, they said, well, you're going to start your vehicle out on this platform of sand. You have a completely vertical carpeted wall that you have to get over and get on the platform on the top. And so I told my son, what do you think is the first thing we did? We came home. Did we start building the car? What did the rover? What did we do first? I told him the first thing you do is you build the platform, because then you could have a platform to test your rover on. And the same thing we do in science, right? If you have a disease, a genetic disease in this case, you create the genetic model. So we went to a model that is easy for us to manipulate. We use a zebrafish. And then we can, what's great in zebrafish is that they develop in front of you. It's not inside the belly, so you can see it happening. You can make multiple type of mutations. So, you can take out 1 copy, 2 copies, and in fish, they actually have 4 copies of this gene. So you can modulate how much you can take away so that you can mimic that phenotype. Because keep in mind, in people with the mutation, sometimes they have the liver problem, sometimes they don't. And you can imagine if you try to do the same thing in mice, it's not that easy. In fish, we're able to just control the dosage of this gene that's affected jagged, and we can model the severity, and we actually made it in a way that it's really severe and consistent, and then ask how that happened. Because sometimes you have to take things to the extremes to understand how things work, right? And so that's what we did in fish, and that's how we initially figured out that actually this gene is important for making that cell type. Before, people thought the gene is only important for changing the shape of that cell, the morphology of the cell. But now we know that it's actually required very early to create that identity itself, right? So that tells you that if the cells are lost, the identity is gone. Can they regenerate? And that actually led us to the next question: is can they regenerate, and where do they regenerate from? How do they regenerate? And we found answers to that. Where they regenerate from. Well, first, they can regenerate. Second, where do they regenerate from? And then third, how can we stimulate that regeneration? So we found that actually if you just let that gene come back, which is kind of an obvious thing, right? But not everybody see it that way, right? Because if the gene is gone, people say, well, it's gone already. Even if you let it come back, nothing's going to happen. That was one of the surprises. We found that when you let it come back, it actually can rebuild itself. That's not true in all cases with birth defects. If you lose gene function even temporarily, the defect's done. You're not going to get that limb back or whatever, right? In this case, the surprising thing is if we let the pathway come back, it rebuilds itself. And the reason why it could rebuild itself is because we found the stem cell source that wasn't affected from the loss of that gene. So when that gene comes back, those stem cells will get used. The gene's not there. Stem cells the liver tried to use the stem cells, but they're kind of helpless without that pathway because the stem cells can go into the liver, we found, without that pathway, but they cannot become those duct cells. And so here we found that they need that gene to be active to become that duct cell. And so the question is, how can we do that? So we knew we wanted to do that, and we actually even looked into gene therapy. We tried to connect with a company that does RNA delivery. RNA is basically a way that the DNA is used. The DNA codes for RNA, which then makes jagged basically, or other protein products. But so we looked into that. And many other ways. And then one major thing that we wanted, because we already knew if the pathway comes back, they can regenerate. So we had brainstormed a whole bunch of different ways to stimulate this pathway. And we'll still want to test all those other ways to stimulate it. But one way that a lot of people want to do it is a simple drug. We were skeptical about a drug because this pathway has been discovered probably over 3, 4, 5 decades ago. I would argue even more, even before we knew what DNA was, this pathway was known. Yet there hasn't been a drug that can increase it. There are multiple drugs that can actually decrease the pathway. So it's always easy to kind of destroy things, right? To decrease something. But to increase it, it's actually doesn't exist. Nobody's found a drug to increase it. And so that's why we were so skeptical that there is even a drug out there that can do this. But you have to sometimes give things a chance, and we did, and we found that it works. It works beautifully. So now we are trying to see if it will be useful for Alagille syndrome. And you'll be surprised, some of the parents are so knowledgeable, because with rare diseases, you can't just go to a doctor and ask, in most cases. And some of them do have to go look into it. So as soon as I realized that we can do something about this disorder, I organized the first rare disease day conference specifically focused on Alagille syndrome. That was done maybe 2017. And the reason for that was just because I knew we're hitting at something that we have a disorder that I think we can do something about. So I thought, well, let's gather everybody together. Why isn't there a conference on this specifically focused on the disease? So many people out there are already interested in notch signaling. So many people are interested in the different organs that are affected, the liver, the heart. Let's bring these people together. And that's how, and organizing this, that led to me meeting up with the organizers for the Alagille Syndrome Alliance, which then allows me to then meet, and they themselves, the leadership there, are family members of Alagille, children, and that's how I got to meet up with several Alagille family members. And actually, it was very inspiring meeting them because, in a scientific way, but also in a personal way. The scientific way is more, you realize what is important to them, right? We may find a stem cell source, which is amazing for us, but they could care less. But they should care in this case because, well, now there's a stem cell that can regenerate these organs. So for me, that kind of made that kind of connection. So so on the scientific side, just understanding what they care about is important, and it pushes us to kind of focus on getting to a cure. But even more on a basic level, like what problems do they have? It's not just the liver. There are a host of other problems. I didn't realize the bleeding was such a huge problem. The hemorrhaging or the risk of bleeding is really big. So now my lab also works on that, and we're going to test whether the drug helps with that also. So that's scientifically it helps in that way, and more on a personal, emotional level. It helps you also focus, right? Anytime you come into a lab each day, there's a million different directions you can take. And if you know me, probably one of my biggest problems is I tend to be interested in everything, and I go in many directions. But they tend to come together eventually, and it's useful that way because you kind of get a broader view on science. But you do have to choose your experiments, and if you have two things that you can study on Alagille syndrome, and you think about the families that are going through it, then you can say, well, maybe I should do this because it'll get us closer to the cure, even though it won't get us closer to another paper or another grant. You make these difficult decisions that may not be ideal for your lab and your career, but it does help move us in that direction. So we've made some difficult decisions to try and kind of put more effort towards our Alagille drug work. But that's why it's so important to know the family, what they're going through. It helps, and every time I hear about a family, every time I get a card or note or anything, it reminds me again, let's push this angle, and so that's why it is important to have these connections. And to be honest, most of us in science didn't go to science because we wanted to find cures. We went to science because we're nerds and we like to solve problems. We like puzzles, and we like to find solutions. We like to be creative, and it's the link to families that then kind of help channel that creativity, that curiosity, the solution-based approach to actually doing something about it. I still argue that just doing basic science is really important because sometimes you look too hard for something, you miss out on really interesting findings. And so I think we should go the other way. We should just do really good science and then ask ourselves the question of where does this good science, where can it be applied in terms of human health? And I think that's it's a less biased approach. When you have a biased approach, it skews your ways. Just like our bias that the drug won't work, it slowed us down, right? If we kept open to it, and we end up finding that it works or not. What I try to put out there, especially coming from my lab and seeing what other people are doing, is that even though it is a rare disease, it has a huge place in research. I mean, I don't want to get scientific again, but the reason because I don't want to just say, oh, it's rare, therefore, it's important. No, there's good scientific reason why studying rare disease is important, not just for people with the rare diseases themselves, but it allows us to understand science better, right? Because rare diseases are often due to what's considered monogenic. It's considered to be monogenic, which means that it's one gene that's affected. And so what we have basically is we can study one gene per disease, as opposed to cancer, which is a ton of mutations, diabetes, likely tons of mutations also. So it's really difficult to study those other diseases, but so much resources are putting into those other problems. But what I'm trying to say is, with rare diseases, there's hope in that we know which gene it is. So that's one major thing I want to put across, and the research is not just going to benefit people with rare diseases or that specific disease. By understanding that one gene, for example, now we're studying Alagille Syndrome, jagged, we're figuring out a way to increase this pathway. And by doing that, we're going to be able to help out with many other diseases that are polygenic, due to many problems with many different genes. So all the effort we put into studying rare diseases will be beneficial to many other diseases. So that's why I would advocate to fund and focus more on rare diseases, because in some ways it's simpler, but in another way it will be more applicable, because we know what pathway it is, and once we know, we can then apply it to more complex diseases. Sometimes I think I don't know if this analogy fits, but if you have a scrambled up mess of something, right? Let's say you have, this is an analogy I use, I don't know if it will work here, but like if you have two wires, right? And they're all tangled up, right? One wire is really long, one wire is short, right? You might want to work on the short wire first to take that out, to untangle it. And once you untangle that, that'll make it a little bit simpler for the longer wire to be untangled. Otherwise, you don't have anything to focus on. And to me, rare disease is like that. It's a simpler monogenic one gene disease that we can work out. And once we have the tools to do that, then we can apply it to other more complex diseases.